Hello, dear students. I'm Li Wenjun from School of Basic Medical Sciences, Shandong University. Today, we'll talk about the basic characteristics of viruses. The first human virus, yellow fever virus, was discovered in 1901. Since then, more and more viruses have been isolated and identified. The emergence of new viruses in recent years continues to endanger human health. As we know, the COVID-19 pandemic that began at the end of 2019 has caused worldwide infection and deaths. In addition, the infection of some viruses is closely related to the occurrence of malignant tumors. For example, the chronic infection of HBV or HCV is closely associated with the development of hepatocellular carcinoma, and human papillomavirus infection is highly correlated with the incidence of cervical cancer. Now let's look at the general properties of viruses. Viruses are the smallest microorganisms compared with bacteria and fungi. Most viruses are too small to be seen under the light microscope, and they can pass through the bacterial filter. Viruses have very simple structure. They are acellular microorganisms, consisting of an internal core surrounded by a protective coat of protein called capsid. The internal core contains either DNA or RNA, but not both. Some viruses have an outer lipoprotein membrane called envelope. Viruses must reproduce within living cells because they cannot generate energy or synthesize protein by themselves. Therefore, viruses are obligate intracellular parasites. Viruses reproduce by means of self-replication. During the replicative cycle, numerous copies of viral nucleic acid and coat proteins are produced. The coat proteins assemble together to form the capsid, which encases and stabilizes the viral nucleic acid against the extracellular environment. Viruses are resistant to many antibiotics because there are no targets for antibiotics in their structures. Now let's look at the size and shape of viruses. Viruses range in size from 20 to 300 nanometers in diameter. Smallest viruses, parvoviruses, virion size are approximately 20 nanometers in diameter, whereas the largest human viruses, poxviruses, have a diameter of approximately 300 nanometers. The shape of virus particles are determined by their composition. Their shapes are frequently referred to in colloquial terms, for example, spheres, rods, bullets, or bricks. But in reality, they are complex structures or precise geometric symmetry. Most viruses that infect humans are spherical. Now we'll talk about the structure and chemical composition of viruses. Virus particle is also called virion. The virion is a complete infectious virus particle, consisting of a nucleic acid core, a protein coat, and in some species, an external envelope. Virions have a core and a capsid. The nucleic capsid is a combination of nucleic acid core and capsid. Some viruses are enclosed within an envelope. If the virion only has a nucleic capsid, it's called a naked virus. If the virion includes the nucleic capsid plus surrounding envelope, it's called an enveloped virus. This picture shows the structure of the virion. The left is a naked virus, which is made up of a core and a capsid. The right one is an enveloped virus. It contains the core, capsid, and envelope. The spike is a virus-encoded glycoprotein exposed on the surface of the envelope. The chemical composition of viruses contain nucleic acid, protein coat, viral envelope, and non-structural protein. Viruses contain a single kind of nucleic acid, either DNA or RNA, that encodes the genetic information necessary for replication of the virus. The viral nucleic acid is located internally and can be either single or double-stranded DNA or single or double-stranded RNA. The genome may be single or double-stranded, circular or linear, and segmented or non-segmented. The DNA genome of viruses can be single-stranded DNA or double-stranded DNA. Double-stranded DNA is the most common type of nucleic acid in DNA-containing viruses. Viral RNA exists in several forms. The RNA may be a single linear molecule or in several pieces. A single-stranded RNA genome can have a positive polarity or a negative polarity, also called positive sense and negative sense. Positive sense RNA genomes have the same base sequence as the viral mRNA, whereas negative sense genomes have a base sequence that is the base pair the opposite of the mRNA. Almost all viruses contain only a single copy of their genome. The exception is the retrovirus family, whose members have 2 copies of their RNA genomes. Viral genome contains all information necessary for viral replication, infection, heredity, and variation, and is used as one criterion for viral classification. Nucleic acid of some viruses possesses infectiousness, which is called infectious nucleic acid. The capsids are virus-encoded specific proteins that protect the genome and confer shapes to viruses. It's composed of small structural units called capsomere. Each capsomere consists of one or several proteins. The arrangement of capsomeres gives the virus structure its geometric symmetry. Viral nucleic capsids have 3 forms of symmetry. First, ixohedral, in which the capsomeres are arranged in 20 triangles that form a symmetric figure, an ixohedron, with the approximate outline of a sphere. Second, helical, in which the capsomeres are arranged in a hollow coil that appears rod-shaped. The helix can be either rigid or flexible. Third, complex structures. Some virus particles do not exhibit single cubic or helical symmetry, but are more complicated in structure. The viral capsid serves 4 functions: protect the viral genome, provide the structure symmetry of virus, determine the antigenic characteristics of virus, and determine the attachment of virus to the cell. The capsid protein is necessary for naked viruses to initiate infection. A number of different viruses contain lipid envelopes as part of their structure. The envelope is a lipoprotein membrane composed of lipid derived from the host cell membrane and protein that is virus-specific. The viral envelope is acquired as the virus exits from the cell in a process called budding. Furthermore, there are frequently glycoproteins in the form of spike-like projections on the surface, which attach to host cell receptors during the entry of the virus into the cell. Another protein, the matrix protein, mediates the interaction between the capsid proteins and the envelope. Non-structural proteins are found in virions or in virus-infected cells. Some viruses carry protein enzymes inside the virion. The enzymes are present in very small amounts and are probably not important in the structure of the virus particles. However, they are essential for the initiation of the viral replicative cycle when the virion enters a host cell. Examples include an RNA polymerase carried by viruses with negative sense RNA and reverse transcriptase. Some proteins encoded by viral genes in the infected cells can interfere the normal metabolism of host cells, such as the transformation protein of tumor virus. Okay, we have talked about the general properties and the structure and chemical composition of viruses. Thank you for watching and see you next time.